Pressure is on. Okay. So what we did here was we put together a very brief demonstration. We'd be more than happy to schedule one live for you. It is basically, this is the device that was used in this use case. So this is the RAD97, and Jennifer will be talking about it later on. It's quite multifaceted in all likelihood, and just about every site that you're going to go to for your studies, you will have a Masimo device. We have a very healthy partnership with Masimo, and we interconnect a multitude of their devices. So if RAD97 doesn't happen to be the device that you have in mind, Root is another one, and the other devices that Jennifer is talking about. The RAD97 also comes with a blood pressure cuff that allows you to monitor the patient's blood pressure as well as the SpO2, heartbeat, or the CO2. It is a very robust tool. It is designed to deliver the telemetry to the nurse's station or to your PI or to your CRA. But in the context of this use case, our goal was to move this data using this middleware, and it's coming from multiple patients. So these patients are identified in a different way, and that this data needs to, in a very fluid way, make its way over to the EDC, which was Bioclinica in this case. So the middleware is quite robust. Repeatability, something that we talked about. Now think about repeatability, not only saving you time, but think about how much it economizes your validation. Because once you have a connector, and this is the portfolio of connectors that the platform has, so the EMR, EHRs are here, and the Masimo device connectors are here as well. The LAS are a number of lab automation systems. The data comes in from the connector, and then it's fed into data transformation rules, and these data transformation rules become a part of that data stream. So why the layers? Well, the reason for the layers is you need to insulate, and you don't, the actual getting the data, reading it in, you want to insulate that from the actual data transformation tool, from the actual pushing it to the target system. It economizes validation and makes everything faster for repeatability for subsequent setups. So here we have data streams that take data from Masimo and push it over to Bioclinica. And we have those connectors, by the way, are bidirectional. So here are the different data streams that we designed using the designer tool. And here is the end result. This is the Bioclinica EDC. Couple of things that are really important. The Bioclinica EDC's interface does not change in any way. Masimo's interface does not change in any way. That's super critical because you don't want to have to retrain all your data managers, CRAs, PIs. They don't like to have to learn something new. The idea here with the DCT is you want to slipstream into what they have. There is no change to the Bioclinica EDC. Data management magically sees data coming in. So this is a CRF. This is a multi-row table. These tables, they have different features, and Richard and his gang can give you a demo of that. But this is a dynamic CRF, and the reason for that is because we don't know how many different data points are going to come in. This is the data. It's coming in the proprietary format. There was a patient ID. And by the way, how do you match patient IDs from a Masimo device at the hospital over to the EDC where the patient has been randomized? These are important data transformation capabilities that you need to have. So here the timestamp at 10:02 was 120/80. And in this protocol, we want to do periodic monitoring unless there is an exception. So here we have another data point coming in. 5 minutes later, blood pressure is still at 120/80. And here, what we now notice is that there was a significant enough fluctuation in the blood pressure that instructed the middleware to solicit data from the Masimo device. So the Masimo device is constantly feeding data. It's the filtering, right? The filtering is, okay, every 5 minutes, everything is fine. Oh, I saw an outlier and it was only 2 minutes later. So this constant filtering is important. Now imagine that. You now know exactly when the blood pressure fluctuation happened here. Now the same thing with SpO2 or the CO2 or you know, any sort of cardiac monitoring or EKGs and ECGs. So here now it's 128/86. It's stabilized. So we flip back to every 5 minutes sampling. There is another outlier, and the outlier now again triggers that the data that was captured here. Now think about how much time we saved between the CRA, the PI, and the data manager here. The fact that the data magically showed up in the appropriate CRF and the entire plumbing was certified. And that's very, very important. It's one thing to write the code for the APIs, but making sure that it's validated and that it's CFR 21 Part 11 and is compliant with the privacy regulations of where the site is and so on. That is huge. You certainly don't want to just have APIs tied together and data flowing only to find out at the end of the study that you overlooked some major privacy regulation and that the data is all for naught. So the system has to be repeatable. The tools, the monitoring tools have to be something that the sites are accustomed to. The EDC has to be something that your data management likes and has been trained in. You don't want to change any of those. And finally, you want to make sure your aggregation tool in the middleware is fully validated and can repeat the same functionality from study to study.